Good afternoon, everybody. Good late afternoon. So I have nothing to disclose. How do I advance this? Okay. All right, so why do we need to integrate data across scales and modalities to investigate white matter pathways? Also, the intrinsic low spatial resolution of diffusion MRI has limited our investigation of brain white matter connectivity to major white matter pathways, like, for example, here, the corticospinal tract in blue or the inferior longitudinal fasciculus. This limitation in resolution, together with the nature of tractography algorithms to step towards the closer or more similar fiber orientation, has contributed to the conceptualization of white matter bundle as monolithic structures connecting two pairs of remote cortical regions. And this is usually also the approach that we take when we need to reconstruct these tracts or extract them from whole brain tractograms. However, this stands in contrast with what we know from tracer studies in non-human primates, where white matter pathways are instead conceived as conduits of fibers that connect multiple different regions. So we investigated whether having high resolution multimodal data can help us reduce this gap. We focused in particular on the most dorsal branch of the superior longitudinal fasciculus, the SLF-I, here in green. This fiber system lies in a very complex regions between the cingulum bundle here in dark green and the more superficial layers of U-shaped fibers in yellow. This complexity has contributed to generate controversial results in tractography studies in humans, with some studies supporting the existence of long-range connection going all the way from the parietal to the frontal regions, and other studies supporting the existence of only short-range connections from the parietal to the premodal regions, some studies arguing against the existence of the SLF-I at all. So to investigate the mesoscopic connectivity of this bundle, we combined in-vivo diffusion MRI data acquired 760 microns and 1.5 millimeters with ex-vivo diffusion MRI acquired at 750 microns from 2 hemisphere samples, from which we obtained smaller blocks along the superior frontal gyrus that we further acquired at 250 microns in a small bore system. Then for each of these blocks, we acquired even smaller blocks on a PSOT system at 10 microns in plane resolution. We then combined this human data with macaque data, for which we have both diffusion MRI data at 700 microns and tracer data. We looked at the data in both a qualitative and quantitative way. So when we looked at the reconstruction of the SLF using the diffusion MRI tractography data from the 1.5 millimeters, we can obtain the streamlines that go all the way from the parietal to the frontal regions, even up to region 10. However, when we do the same for our ex-vivo data, most of the direct connections that we can reconstruct run from the parietal to the premotor region 6, and then more frontal to that, we observe a series of shorter range connections that go in and out of the cortex, and this is very similar for both samples. If we then zoom in, we look more in particular into these frontal regions using the tractography data from the 250 microns data and from the PSOCT, we see very similar results across modalities with this shorter range connection again going in and out of the cortex. So then if we look at the macaque data, the tractography data, again we observe a similar connectivity pattern with streamlines connecting from region 10 to region 9, 8, and 6, sort of going in and out of the cortex, and then from region 6 we can see direct connections to the parietal lobe. So we selected, for the tracer data, we selected case with injections along this dorsal-medial region. So for example, this case with an injection in region D, we can see terminals in region 9. This other case with an injection in region 8, we can see terminals in anterior part of region 6. In this third case with an injection in region 6, we can see terminals all the way in the parietal regions. We can then trace these fibers from the injection to their termination point and create 3D reconstructions of these fibers. So here is a video, for example, that showed the 3D reconstruction of these fibers. So in light blue, you can see the SLF-I as we would find it on an atlas. In pink here is our injection, in this case in region 9M, so in frontal part. And again, in pink, you can see the fibers leaving the injection regions and entering the SLF. The fibers that enter the SLF are here shown in green. And then we can see also in light green, you can see fibers leaving then the SLF to reach cingulate regions or more lateral regions. We can then also map the terminals here will pop up in brown. And you can see that most of the terminals are located actually in neighboring regions with some other terminals in anterior part of region 6. Here there's other two cases. So this case has an injection in region 6, and again the colors are the same. So we can see fibers projecting frontal to region 9, and other fibers projecting posterior to the parietal regions. In this last third case here, as an injection in the more posterior region, so we're actually moving posterior to anterior in this video here, and again, we can see fibers leaving injection, entering the SLF, running within what we consider the SLF-I, and then not reaching all the way to the frontal regions, but terminating in region 6. And the same pattern of connections of fibers leaving the SLF to join the cingulum bundle or the corpus callosum on one side and more lateral regions on the other side. So we quantified this within a structural connectivity framework where we looked at the connections between these dorsal-medial regions across macaques and humans. And we compare results for using two different masks, one that would allow streamlines to course through the SLF regions, but also through the cingulate regions, and one that would allow streamlines to only course within the SLF regions. So the regions in this matrix are organized in an anterior to posterior gradient. And so if we focus on the lower left quadrant of the matrix here, we can see that when we allow streamlines to run through the cingulum, we have a higher number of long-range connectivity than when we allow streamlines to only run through the SLF regions. And also that as we increase, or as the resolution decreases, the number of long-range connections increases, as well as the regions that show long-range connectivity. So to further investigate this, we also downsampled our data to match the lower resolution of our in vivo human data, which is 1.5 millimeters. And we can see that as we downsample the data, again, the number of long-range connection increases, as well as the number of regions that show long-range connectivity. So just to conclude, by integrating data across multiple modalities and scales, we could show a higher level of complexity for the SLF-I connectivity that is similar across species and modalities, and that shows both long-range connection and shorter range connections, particularly in the frontal part of the SLF with fibers going in and out of the cortex. And this complexity is lost in low resolution diffusion MRI tractography, where these different connections sort of get pulled together and result in a long direct association bundle. With that, I'd like to thank my collaborators, particularly Suzanne and Marina, for helping me out with the tracer data, our findings, and all of you for being here so late. Thank you.
Thank you very much. So we have some questions from the audience in the app. Could you please describe the ex vivo diffusion protocol used in terms of shells, sampling schemes?
Ooh, that's a lot. So for the hemisphere data, that's actually a steady state acquisition. So in that case, we have only one shell, and it's not a very high B value. And for the 250 data, we actually acquired that with on the full grid. So we have 515 volumes, up to 40,000 p value, and then we resampled that on shells. So it's a multi-shell protocol.
Thank you. How do these short connections, a question from the audience, how do these short connections relate to the prefrontal U fibers described by Katani and others?
So I actually tried to exclude the I tried to look at the short-range connection, excluding the very superficial layer of the U-shaped fibers, because we don't really want to look at the U-shaped fibers, but to investigate whether there were short-range connection within the SLF.
Anything else? I have a question. Well, maybe some original study of the tracer by Schumacher and Pioneer, maybe according to their book, at least in the cartoon level, it looks like maybe SLF-I is described like coming continuous bundle. I'm not sure how much it's aligned with actual raw tracer data, but do you have any comments about relating original Schumacher and Pioneer book, just description how much these are aligned with the actual observations?
So actually he doesn't describe, if you look at the illustration, there is actually both long connections going all the way to 9 and short connections going to 6, and other relays in the premotor areas too. So it's not actually one. And it is possible that, there's both systems that run within the same pathway.
Thank you. So maybe final question, which atlas template was used for the spatial longitudinal fasciculus?
Oh, there is no atlas used. So the regions are actually placed manually. When I mentioned the atlas, even for the macaque data, we usually just define the borders. In this case, comes from Shaman. But there is no actual atlas involved in the tractography data.
Okay. So the time up, and thank you again for the speaker.
Thank you.
So let's move on to the second speaker in the session. Could you show the slide?